The locus coeruleus is a deep brain structure that regulates behavioral arousal, attention, and cognitive performance. Its activity can be recorded indirectly by measuring the pupil size.
This approach allows the verification of whether stimuli that boost cognitive performance, such as trigeminal stimulation, modulate LC activity as determined by changes in the pupil size.
Experimental evidence suggests that neurodegenerative diseases are associated with locus coeruleus dysfunction. The present approach will pave the way for the modulation of trigeminal input to the locus coeruleus for therapeutic purposes.
Demonstrating the procedure with Tommaso Banfi will be Vincenzo de Cicco, a professional physician and odontologist.
To perform the experiment, a commercially available piece of chewing gum, referred to as a soft pellet, and a silicone rubber pellet, referred to as the hard pellet, are needed. A tangram puzzle must also be prepared for the haptic task. For Protocol 1, provide a paper displaying 3 10x10 numerical matrices to the subject and ask the subject to sequentially scan the matrix lines while using a pencil to tick as many target numbers indicated above each matrix within 15 seconds to measure the subject's baseline cognitive performance. Then manually evaluate the performance index, scanning rate, and error rate. To measure the baseline pupil size, seat the subject at an optimal working distance of 56 mm from a corneal topographer pupillographer and acquire single, separate camera shots of the